Well, good morning, good afternoon, everybody. This is Arthur Brandwood from Brandwood Biomedical, and welcome to this February 2019 edition of our live webinar series. Today, we're going to be talking about biocompatibility, again, biocompatibility challenges for 2019. I'm joined here with my colleague, James Morrison, to talk about changes in the ISO 10993 series of standards. James. Yeah.
G'day Arthur, how are you? And g'day viewers, welcome to the webinar for 2019. Allow me to introduce your panelists today. So I've been a member of TC 194 for a number of years now, and I'm head of the Australian Technical Delegation. My particular expertise is in respiratory devices, and I was quite active in the development of ISO 18562, which is a series of biocompatibility standards for breathing gas pathway devices. So that includes ventilators and the like. I'm currently engaged in the revisions of Part 17 and Part 18 through the ISO Technical Committee, and I have a degree in materials science and experience in toxicology. Arthur is one of the longest serving members of the ISO Technical Committee, and he was part of the working group that drafted the new Part 1. Arthur's got a biomaterials and regulatory background, and he's got 30 years experience with material tissue interactions. So that's who we are. So without any further ado, why don't we begin, Arthur? And how about you tell us why and what the recent revision was all about?
Well, thanks, James. So Part 1 was recently revised, published just 6 months ago. What was that all about? Well, the foreword of the document actually talks about what's changed, but really there's a couple of big changes. The changes have been about cleaning up the annexes, yet again revising Table A1, which is one of the most important parts of the document, but is always been poorly understood, I think, and we've made further efforts to make that clear. So the main changes in the document, Table A1 has been updated, and we'll talk about that in some detail in a minute. A lot more emphasis on understanding material, characterizing the chemistry and the material itself, or materials in the device, making this more and more a risk management guidance. 10993 is biological risk management exercise, and the new Annex B is all about that, for example. And a couple of other smaller changes, such as defining clearly what transitory contact is and how devices which have very limited contact don't need much biocompatibility evaluation of at all. What's all that going to mean? Well, manufacturers need to understand these changes. You need to be better informed, plan and understand the risks and focus on them. This is a risk management document. Risk management is about learning, defining what things matter and putting your attention there. There is another side to that, of course. It also allows you to justify what things are not really very important and put less effort into those. And so you can really focus your attention. Regulators and notified bodies really need to understand this as well. There's a lot more in Part 1, and there's a lot more coming in some of the other parts. It's not about just increasing workload. It's about the risk management model. It's about knowing what's important, what it is you're dealing with, and focusing on that in your reviews of technical submissions, not just having a very long list and have we done all of these things. Consultants. If you're engaging with consultants or advisors on this, you need somebody who really gets that risk management piece. You want to be helped by somebody who knows how the standard works, can explain to you how to apply it, and can guide you into what activities you need to undertake, because you can't do everything here. You need to be focused and you need to pick on what matters. And so if your consultant just wants to apply a large checklist, then maybe you've got the wrong consultant. If you've got somebody who really understands the risk management framework here, that's the sort of group you want to go with. So a lot going on, James. maybe you'd like to talk about what's been going on in terms of the different changes going on in parallel.
Sure. So there has been a bit of a push to revise a number of standards simultaneously, and those sort of emphases there were on the revision of Part 1, again, as Arthur said, to reinforce the risk management process ideas around biocompatibility, and then to revise Part 18, which everybody thinks of as being extractables and leachables, which is really about tolerable intakes, not tolerable intakes, but chemical characterization. And that's to actually empower or to provide data for risk assessment processes. And then Part 17, which deals with allowable limits, is turning into a risk assessment guide or textbook almost, and it's going to be renamed toxicological risk assessment. So that's what the changes were all about. So why do them in parallel? Well, it actually doesn't make sense to require risk management in Part 1 without providing good tools to enable it to be conducted. So there was a push to simultaneously revise those three parts of 10993. But the reality is that that didn't happen. Part 1 was completed and published in August last year, and that in itself was perhaps miraculous in that it was so quickly achieved. Part 18 is due out late in 2019 or perhaps early 2020. The bulk of the work is completed, and we're now going through the ISO process that will lead to publication. Part 17 will run third. Perhaps it's not inaccurate to say it may run a distant third. It's probably unlikely to be published for a couple of years yet. And largely this is due to the technical complexity of these documents, the novelty of the content, and the need to achieve consensus. Remember that international standards are consensus documents.
I think, James, the experience, particularly with Part 18 and 17, there's been a huge amount of debate in the committee so far, and certainly not there yet on consensus. There's a lot of arguments over some of the approaches and amongst the experts themselves.
Indeed, and we experts are continually learning. And as experts bring new factors to the table, then they need to be taken into consideration and see if a consensus exists and whether the information is fit to be put into the standard. So what else is in progress? Well, of course, there is a continual revision program for all the other parts of ISO 10993. And so there are some notable things going on there, particularly to do with in vitro test methods for things like irritation. And there are other revisions going on as well. So it's a fairly full program, and there are a lot of things going on.
And a lot of that's driven by alternatives to animal experimentation, more sophisticated testing, and so on. We don't have time to go through all of that right now. Before we move on to the next slide, I neglected to say at the beginning, of course, that we accept questions during these webinars. So if you look at the bottom of your screen, you've got a lot of control buttons. Scroll the mouse down, you'll see a bunch of control buttons. One of them says Q&A. That'll pop up a little screen for you separately, and you can type in a question, and we'll take those questions at the end. So let's carry on. So
thanks, Arthur. Look, Part 1 now aligns mostly with FDA guidance, and I noticed that in the last month, FDA have added the new part of Part 1 to their list of recognised consensus standards. So does this mean that everyone will have to do more testing?
So what you're saying is the table A1, I think it all hinges around table A1. Table A1 is, of course, that big matrix at the back of the document, which lists all the possible endpoints that you might want to consider according to the exposure and duration of contact of your device. So for more invasive devices, you tend to need to think about more things. The challenge with Table A1 is it has in the past been used as a checklist for doing tests. and the perhaps naive user would go straight to table A1, say, oh yes, my device is externally contacting a certain period of time. That means I need to do these tests, and then go and commission a lab to do a set of tests, and that was it. That's not how the standard's meant to be used. What those ticks in the boxes in that table are about is what you have to think about for your particular device. So it's giving you a guidance as to what matters. FDA has always been visibly more conservative about that, wanted people to think about more things than were ticked in the standard. And so FDA have had in their guidance a modified table A1 with some extra boxes ticked. That's been misunderstood. FDA's position has always been, we want you to think about more. And I've had discussions with some of the leadership at FDA about this, and they say, look, no, that doesn't mean they want you to test more. What they want is that your submission needs to address all of the boxes they've ticked. They want you to talk in your submission about each of these things and explain whether you have tested or whether you don't need to test, and cover all of the topics. So doesn't mean that everybody's got to do more testing. It doesn't mean that FDA has won an argument. I mean, it's not FDA winning an argument. That's the wrong question. It's understanding what FDA's viewpoint is, that really they're saying, you know, we want you to think more broadly and justify your approach. So you've got a much more comprehensive standard now. It's going to take a broader approach to work on. So let's move on to the physical and chemical information, which is really the big new change in the standard. There's a lot more about that. So James, you want to just talk us through about the new emphasis on?
Yes, thanks, Arthur. Really, the drive for physical and chemical information is about knowledge of products. Now, you might note here that I've used a word that's not usually used in terms of biocompatibility, and that is products. So really, it's a thought about what is placed onto the market and how much is known about it. Perhaps to our detriment, the medical device community does not actually have a very great knowledge of what their products contain or are constructed from. There are an awful lot of device manufacturers out there who just think, oh, the device is made out of polypropylene. Well, that's a generic polymer name, and it may in fact involve an awful lot more than polypropylene. And so this is about, what do we know about what we're putting onto the market? And of course, the reason for that is we want to have a degree of control. If we want to mitigate and manage the risks associated with medical devices, then we have to be able to control those products. And so really that is what this emphasis is about, so that everybody understands what's in a particular product and device. And so clearly that's going to have some implications for the manufacturer. And indeed, in table A1, the very first column now is get your physical and chemical information for all devices. And the rest of the standard backs this up, basically saying, do this step first. Make sure that you find out or that you know or that you're able to control the chemical and physical characteristics of the device before you begin any other parts of the biocompatibility evaluation. Again, know and control your final composition. But you only have to do as much as the circumstances require. So if you have a relatively low risk device, then you may not have to do anything like the sort of work or know the sort of information that you would have to for a permanent implant device in, say, neurological tissue. So you have to do what the circumstances require. And that, of course, will involve the judgment of the manufacturers and a rationale for why a particular approach was taken. And of course, every rationale needs to be written down so that it's accessible to regulators and notified bodies and anybody else who's an interested player. So this is about knowing, saying what you know, and say why you think that's okay.
Mm-hmm. And the changes in Part 1 particularly, and the new chemical characterization content have I think led to a boom in laboratories advertising new services. We can do more chemical characterization. We can do more of this testing. How do you know about a lab? How can you be sure about which is the right lab? What do we need to think about in picking a lab?
Well, it seems that every laboratory, whether they're a CRO servicing the medical device industry or just other laboratories out there, are offering E&L, extractables and leachables as a service these days. And the first thing I need to say about getting a laboratory to do something for you is that you have to work with them. This really is something where you know about your device, but you don't know how to get the chemical information about it, and they don't know about your device, but they might know how to get the chemical information about them. So let me say at the outset, this is all about collaboration. You must work with your laboratory, and vice versa. They must also work with you. Because the big question is will they do what you want, or will they only do what they want? So laboratories, I think, are going to have to learn how to be flexible, and they're going to have to be responsive to what their customers are saying to them. In the past, this has not always been the case. Let's hope that there is a bit of a shift in mindset on the part of everybody in the industry, so that we can begin to do this and do it sensibly.
So what does that mean in practice? So tailoring the testing, understanding exactly what...
Yes, that's right. And, one of the questions that's asked is, well, look, what's the standard test that I have to do? Is there just a standard assay or something that I might do? And the bad news is that there are no standard tests, because there is no standard medical device. There are 10 different ways of doing a cytotoxicity test, for example, and you would choose one of those methods according to the type of device that you had. And the same applies when we talk about physical and chemical information. And the new Part 18 is going to have a lot of very helpful flow charts and indeed tables of options to help in the selection of methods. Because, of course, you will have to use very different methods if your device is a ceramic to if your device was a metal, and again, a different set if your device was made from polymers. So there will be some very good toolkits, if you like, in the new Part 18. But you always have to remember why you're doing this and scale this appropriately. So you just have to do as much as the circumstances require. I really can't emphasize that strongly enough. This is not supposed to impose a great burden on everybody. It's rather an opportunity to make sure that we know what's in the product and what that means in terms of biocompatibility.
Mm-hmm. It's interesting. I've seen beautiful websites from some laboratories that offer a quick Q&A about what your device is. You ask some questions about your device, and then it spits out a list of tests that they can do and the reference numbers for their tests and a quote. Isn't it a little bit more complicated than that?
Well, Arthur, let me say, I think that's a bit of a cheeky question. Clearly, you need to know what you need as a manufacturer, and labs can help you decide how to do it. But what you need, it might be something that you need to negotiate with the labs. And let me say that it's always better to have personal contact with these people rather than just use the online menus. So when you find a lab that looks like it's offering the sort of things you're interested in, for goodness sake, get on the phone or write them an email, enter into an interactive process with a person, not a computer, and begin to negotiate how you can work together. You're going to have to refine processes with the laboratory. It might take a little bit of time for them to be able to demonstrate that what they're doing for you is actually effective. And you might need to validate some of those processes simply so that you can say, well, when we did this test, we knew that the test was meaningful and significant. Ultimately, it'll get less complicated as manufacturers, and indeed the labs working with them, have more experience. They'll work out how to do it and streamline it, and it will become less complicated.
You're going to need to work with the technical experts on you, and the good labs that's what you'll always get. You'll get direct access to the technical experts.
Clearly.
You're not just going through the salesman who's going to sell you a list of tests.
That's right. But your first point of contact will be the sales and marketing arm of a corporation. Once you explain what you want, they should very quickly be passing you on to talk to some people with some good technical skills. They might not be the technicians who do the work, but they'll know something about it. And so we need to be wise. We need to go into this with our eyes open and not just throw it over the wall.
So there's a whole lot more now. We've got all this chemistry. We've already had all this expensive biology that everybody does. Now we've got chemistry on top. Isn't it going to just cost a whole lot more?
Well, I think there are three ways to measure cost. There's the cost in terms of time, the cost in terms of money, and the cost in terms of animal. The animal welfare burden is not to be ignored in this equation. Very often, we only think about cost in terms of money. I think that the reality is that for a lot of device manufacturers, doing the chemical characterization and the toxicological risk assessment will in fact ultimately reduce the amount of time, money and animals that are expended in the process of establishing a biocompatibility. However, that's only going to work if you restrict what you do to as much as the circumstances require. If you want to, somewhere we have everything you can afford to pay for. A laboratory will continue to spend your money while you continue to give it to them if you're a manufacturer. And you don't want to do that. You want to do as much as the circumstances require. Ultimately, it's going to be cheaper, but there will be a steep learning curve, I think. There shouldn't be, but I'm afraid that there will be quite a steep curve.
And chemistry can be done more quickly. It's these days more sensitive in terms of finding out what's in there, and then you can go to the literature and the toxicology of what that's present. But learning to do that properly is going to take some time and some adjustment, I think.
Yeah, that's true. And also, there are a number of biological endpoints that won't be able to be addressed using the chemical and physical characterization information. You will still have to perform some biological tests. I'm fairly certain of it.
Look, this is all very complicated. I'm going to dig a bit deeper and say, you said, make sure the labs do what you want them to do. How do you know what you want to do?
Well, the first thing is that you need to have a plan. Very often, biocompatibility has historically been done without any plan at all, and that's a reflection of the checklist mentality. These days, you need to have a plan, and the first thing on your plan is going to be work out, of course, what sort of characterization your device has in terms of body contact and contact duration, and then begin to make a plan of how you will evaluate it for biocompatibility. You have to think about biocompatibility at the beginning and in the middle of the product development process. You don't just think about it at the end. It's not a validation exercise. You don't just do the biocompatibility testing at the end of your program and say, well, thank goodness that worked. You've actually got to think about this right at the beginning of your PD process. So you've got to have a good plan.
Biocompatibility is a design input.
Yes, it certainly is. And indeed, how you evaluate for biocompatibility should also be a design input. Again, let me make the point that you need to collaborate. So you need to make sure that the product development process and the cross-functional team is empowered to make decisions. Very often, product development processes are not nearly as empowered to make decisions as they need to be. And there has to be a broad spread of technical expertise available to the XFT. And even if you have to subcontract some of that expertise, you still need to make it available. I've got a little list here of the sorts of skills that I think are going to work well. Clearly, you're going to need to involve the design engineers in this. That may be a bit of a surprise for a lot of people thinking about biocompatibility. Why on earth would we need to talk to the designers? We're just trying to establish the biocompatibility, not the shape of the thing. But of course, biocompatibility hangs on a number of factors. And this is not just chemical considerations, it's also physical considerations. So clearly your design engineers need to be involved. Material scientists, if you've got them, that is, not as common as I would like them to be, but that's my own professional bias. But certainly you need people who know about materials or they know enough about materials for what you're trying to do. You would need to involve your manufacturing engineers because the manufacturing engineers will be able to tell you, well, when we converted this material into this component, this is everything else that we did to it at the same time. So we used a mold release agent, we used a lubricant when we cut the steel, we plated it, we coated it, we anodized it, everything that they did in the process. So that needs to be factored in because it's all part of the final product that we're evaluating. You're going to need some analytical chemists. That's clear because this is about chemical characterization. You'll only need as many of them as the situation warrants. And of course, you'll need toxicologists because all this is about a toxicological risk assessment. And again, you'll only need as much toxicology help as the circumstances require. So there's a fundamental difference in how much expertise you need according to your device type.
So we're talking there about expertise in how the device is designed, how it's put together, and then how people interpret the chemistry and the biology at the end.
That's absolutely right, Arthur.
And how do you make sure they're doing it?
Well, the best way is to hold their hand. That is, you must collaborate. Look, we all need to get smarter. We're all going to be on a learning curve here. Everyone in the industry is going to have to learn how to do this, and if we learn together, that will be good. You can't just throw things over the wall. That was the way that we used to do things when we treated Table 1A like a checklist. I think we've moved beyond that now. And it's really going to be a case of work together. And everybody has to learn how to not only to work together, but they have to learn how to explain what they're doing and what they need and why they think it matters. You're going to have to teach one another. And that way, as I say, we'll get smarter and we'll be able to ensure that what we have is, in fact, what we need to have.
Good. Well, thank you. It's time to take a few questions, I think. Firstly, just a couple of technical questions about the webinar. This webinar, just to remind everybody, is recorded. Everybody who's registered will receive a message from us after the webinar where you'll be able to listen again to the recording. You'll be able to get a copy of the slides. And so all of that information will be sent through later, and we put it on a podcast as well. But we also have some questions here. We've got some that were sent to us in advance. If you're a regular listener to our webinar series, we're quite happy to have people email us in advance on help@brandwoodbiomedical.com if you want particular issues addressed in the webinar. So the first one, we've got Sarah from the US on biocompatibility and 3D printing. How do you do biocompatibility for 3D printing, James?
Well, that's a really good question, Sarah. Let me say that this has been thought about for quite some time. And I do know that US FDA has some guidance around 3D printed medical devices, and it does touch upon biocompatibility. But the reality is that 3D printing is just another manufacturing process where you convert a raw material into a finished shape. So really there's no difference for doing biocompatibility for something that's made in that process to something that is, say, injection molded. You may need to take into consideration some things like, is the process actually a reactive process? Are you curing something when you 3D print it, or are you simply fusing it together? You might need to take into account the purity of the materials. I know that a lot of the processes that involve powder beds recycle the powder a number of times, and so you might have to have some really good controls around powder recycling if that's what you're thinking about. And also there would be residuals from any sort of process. There would be post-processing. In my experience with 3D printed objects, there's an enormous amount of manual post-processing that you have to do to turn the printed object into a final product. So that would also have to be taken into consideration. So really good process understanding is important for 3D printed objects, almost to the same extent that understanding the materials chemistry is.
Thank you.
Now, one of the difficulties of doing webinars in Australia and providing them to the rest of the world is that it's very early in the morning here and the security system has turned the lights out on us. So we're struggling to read. So Arthur, can you actually read that next question? I'm sorry, I'm struggling here.
I've got a list of questions that were emailed in advance. So yeah, I'll launch it. So Vim from the Netherlands asks about, can we explain the difference between the new and the old versions of 10993? Well, I think we've covered that. That's why we added some content to the webinar, Wim, but thanks very much for the question. 7 changes, speaking to changes in the main table, into additional chemistry, and a couple of things like transitory contracts. So as we covered off in the webinar. Henry from the UK, from the United Kingdom, asks, what about the medical device regulations and the general safety and performance requirements with GSPR, the general safety and performance requirement 10.1H, we're getting quite specific here,
yes.
Means that there has to be chemical characterization. It's written into the new European regulations. Do you want to just talk a bit about that?
Yes, well, thanks, Henry. Again, sensible question. I'd like just to quote that part of the MDR because I think it's pretty important to make sure that we understand what we're dealing with here. So it says, devices shall be designed and manufactured in such a way as to ensure that the characteristics and performance requirements referred to in Chapter 1 are fulfilled. And particular attention has to be paid to, Part H, the confirmation that the device meets any defined chemical or physical specifications. So this is actually about verification. So if your specification defines a chemical or a physical characteristic, then you have to validate for it. That's really what this says. So does that mean that you have to do chemical characterization? Well, maybe not. What does your specification say? If your specification says that it needs to be made out of 316L stainless steel, then it may be adequate for you to get the certificate of analysis for that lot of stainless steel that was supplied to your manufacturing centre and check that it does in fact meet the specification requirements for that alloy, and then you're done. You have confirmed that the device meets a defined chemical specification. And so you've done it very simply. You don't have to do a test. You have to evaluate for these things.
So if you've got a requirement for a maximum for a certain trace element or something, then you could get a certificate of analysis from a material supplier that says we've analysed and it's below the...
Yes, you certainly could do that. Yeah, that's right.
Okay. Moving on, Jennifer from Canada asks, what are the expectations for retesting when the standards change, and how does that differ from country to country? With all of this change in standards, do you have to go back and repeat the testing?
Well, actually, no. Really what you have to do is you have to do a gap analysis to understand how much has changed between one version of the standard and the next. And very often, when a standard is revised, nothing is taken out and very little is modified, something is added. So often it will be the addition of a new test method which is equally applicable. So in the recent revision of the standard for irritation, they added a test which was an in vitro irritation test. They didn't take anything else out. So if you had previously done testing using an in vivo method, then that method is still included in the standard and you would still be compliant.
Let's talk about a specific example there. I remember working with a manufacturer a little while ago who had been asked by their notified body to justify the fact that one of the animal tests done on the device didn't use the specified number of animals that's in the current version of the standard. I can't remember which one it was, sensitization or irritation test. Standard had been modified, asked for a slightly larger study design. And they said, we've been asked to justify why, because we did this 20 years ago, and we didn't use as many animals back then. And the answer was quite simple, actually. Well, yes, the standard had changed and required a slightly larger study design, but your old tests produced all completely zero grade, so you were way down the bottom end of safe, and it was highly unlikely that adding another animal would have made a material change to the outcome. But apart from which, you've got 20 years of the product in the marketplace with the best testing of all, and that is use in actual patients. Go look at that data set, and if that shows that you're not seeing a particular endpoint you're looking at, then that's actually better evidence than an animal test, which actually is a proxy anyway for the human use. So go look. So the answer, I think, Jennifer, is yes, you might have to justify it, but it's rare that you're going to need to retest. It would be very rare. Look at the history of the device and justify why you've got enough data now that the device is safe. As for countries to country, that's more about when you're doing the primary testing, when you're testing for the first time. You do need to be aware that some countries have slightly different protocols for things like extraction methods and whatnot. One of those countries being Japan, for example. Been some interesting work done by some of the experts on the committee. The Japanese variants on the test do seem to be actually a little bit better. So be aware of that when you're going into different markets. Careful about what the local standards are. Look at the national standards for your market and check that they haven't been modified.
So, Arthur, we have a good question here from Julio in Deutschland, and he's asking us about risk management processes and the necessity of having a signed biological evaluation plan and not just a biological evaluation report. Well, when's this appropriate? Well, Julio, I have to say I think it's appropriate all the time because ISO 14971 requires a structured biological evaluation plan. So at the outset, you have to plan what it is you're going to do, and that would need to be reviewed, and everyone would have to agree, yes, that's appropriate, and then you go ahead and do it. If you don't have a plan, well, how do you know what to do? And how do you know that when you've done it, you're finished? So really, having a plan, having sensibly sat down and thought about what do we need to do, and agreeing that that's right, is absolutely important here.
And you said ISO 14971, of course, that's the risk management standard. So 10993 is a special case of risk management.
Yes.
Risk management for biological safety. And both of the standards have explicit clauses in them that say there shall be an a priori plan. You need to map out your exercise first.
Yeah, that's right. And as Arthur alludes to, ISO 10993 is actually a specific example of what we're instructed to do in 14971. It's how to do risk management for biocompatibility. One maps to the other. And indeed, the new Appendix B in Part 1 of 10993 is largely built around 14971, and there are the same pictures in it even. So it really is very much, this is how you do this, and fit in with the rest of the standardized world.
And there's a separate question here from Sam in the United Kingdom who wants to ask their opinion on the new Annex B about incorporating biological safety into risk management, something some people struggle with. I have to say, I think my opinion of the new Annex B is it's a marvellous piece of writing.
Yes, but Arthur, you wrote it.
Didn't write all of it now, but I did have a lot to do with the previous technical report that then was put in, and there was a lot of other people had work in modifying it and improving it. But no, what that Annex B is about is a marriage of 10993 and 14971. So you need to think about it in those terms. You need to think about, okay, what that annex is trying to tell you is how you're doing your biocompatibility evaluation to fit in with the broader risk assessment activities that go with your design inputs for risk management.
Indeed. Emmett from Ireland has asked us, when do we expect Europe to harmonise the new Part 1? Well, Emmett, harmonisation in Europe usually takes about 6 months, and so we're coming up to 6 months since the document was published in August. However, it might be a bit delayed this time because there's a lot of things going on in Europe at the moment with MDR, and that may get in the way. But I think that it won't be too much longer before we see the European harmonised version come out.
We've got a number of questions come from our audience here. So we've got Shravan, who says, it seems that the move is shifting from animal-intensive toxicology tests to more chemistry-based tests, which are more quick and more reproducible. Is that a fair comment?
Yes, absolutely. The drive here is to have less reliance on tests that involve animals. We've actually had that drive for quite a while as we've been driving towards in vitro testing rather than in vivo testing. But now we're taking it a step further and saying, instead of doing tests, and some of these tests run for a very long time on animals, then let's do them a different way and let's use tox risk assessment.
Okay, question from Bo. It says the top 3 US contract labs currently taking 6 to 8 weeks for a basic E&L study, extractables and leachable study, and that's $20,000 to $30,000, not including any toxicological risk assessment that goes on the back of that. What do we see outside of the US? Is this a particular US thing, or are we seeing it globally? I don't know how to answer that.
Yeah, I would say that the turnaround time might be a little longer than would be necessary. So I think that there's probably a bit of fat being put into that by the service providers. I would have said that perhaps 4 weeks would have been a more realistic turnaround time based on my own experience. However, my own experience has been that I worked with the lab for many years to refine and validate processes. And so once we had it up and running, then that was the sort of turnaround time we could achieve.
There's probably a lot going on, a lot of new customers coming into the...
Correct. And perhaps that turnaround time involves, the ramp of working out what a manufacturer needs and the best way in which to do it. So there may be a lot of negotiation time in that. As for costs, that again is very hard to answer because it would depend on the availability and the type of analytical instruments and people to run them. So that's a very difficult question to answer, and it would differ, I think, for every manufacturer's requirement. So no comment, I'm afraid, Bo.
I would make one comment, and that is most of the big labs are in the US and in Europe. We're seeing more and more in places like India and China as well as contractors, but certainly the big centre for the testing labs are still in the US and Europe.
Yes, I think that's right, because I think that the CROs who have historically done the animal and in vitro testing are the ones who are picking up on this. But of course, there are very many other analytical chemistry labs who are able to do characterization work. And again, if I can speak from my own experience, I've actually used a laboratory that was an environmental chemistry laboratory, and their core business was doing environmental monitoring for contamination. That happened to be a very good fit for the sort of analytical chemistry that I needed for medical devices. So although the industry is used to working with the biological testing CROs, there's in no way a requirement to continue to do so if you can get your chemistry needs met elsewhere.
A really good question here from Martin. Martin says, is the expectation that the chemical characterization is done prior to biological testing? Because that, in theory, could significantly extend the duration of the project if you're doing the testing on final devices. What order do you do it all in, and how do you manage to not let your project grow out
Sure.
for years?
Yeah, sensible question. Characterization does need to be done first. The standard is quite clear on that matter. Do your characterization first. Again, we have to bear in mind that you do as much characterization as is necessary. And if you've thought about biocompatibility at the beginning and in the middle of your product development process, then this shouldn't be a massive impost. And toxicological risk assessment based on chemical characterization is going to save you months, if not years, in animal testing at the end of the validation phase. So although it seems like it's an impost, if you get these things in the right order and at the right time, you will come out even, if not in front.
Can you be doing chemistry before you've got to final device stage?
Well, you certainly can, because you can make some representative components. So you might still be thinking about, well, what are the final radii on this component, but you can be doing a representative component chemical analysis. So yes, you certainly can.
So testing something that's manufactured from the final materials is manufactured using the same processes. And I suppose if you do introduce a late change in the manufacturing process or whatever, then you may need to do a bit of supplementary chemistry, but
sure,
or you may be able to justify that the change.
That's right.
Yeah. Okay.
And there is a lot of evaluation that goes on here. You need to put the options side by side and say, are these the same? And if they're not exactly the same, how much does it matter? So evaluation is pretty important here.
Last question from Abby. Hi, Abby. Abby's over in Toronto. We know Abby well. We know Abby well because she used to work here and she's gone off to Canada. Any idea on when the new Part 1 standard will be recognised by FDA and added to the EU list? Well, we talked about the EU one, but you mentioned earlier, I think, that FDA have now actually recognised the Part 1.
That's right. They've recognised Part 1, and it's a very interesting recognition because often FDA will only partially recognise a standard, and they always nominate the bits that they're not going to recognise and the reasons for it. And in the case of the new Part 1, FDA has recognised all of the standard except the Table A1. And the reason for that is because Table A1 does not agree with their current published biocompatibility guidance. They have their own Table A1 in their own guidance. I am sure that this extent of recognition is simply because FDA guidance takes a long time to review and change. And I wouldn't be surprised if in the next while, and I'm certainly not going to put the squeeze on anybody in particular by saying how long that while might be, but in the next while, I think we will see a revision of FDA's biocompatibility guidance where the Table A1 is in fact harmonised with the table in ISO 10993, and then the recognition will become full. That's my projection of what will happen there.
We'll wait and see. Certainly FDA was very heavily involved in the redrafting of the new standard that a lot of FDA representatives.
They were indeed.
Okay. Well, thank you, James. I think we've come to the end of our questions, and we're just coming up to the end of our time. Thank you for all of the questions that have come in, both in advance and from the listeners to the webinar. We'll be back again next month. Meanwhile, look out for an email from us, which will give you a link to the recording of this webinar. You'll be able to download a PDF of the slides, and you'll also be able to download an audio for a podcast.
Of course, and if you think of a question that you haven't thought of during this session today, then please send us an email. You can see that our email address is here on this slide, and there are lots of other ways that you can engage with us. We have all those other sorts of tools. We have Twitter, we have LinkedIn, we have YouTube. We've got a Facebook as well.
Yeah, we have a Facebook page.
Please like us on Facebook.
and the webinar will also be posted to our YouTube channel as well. So with that, thank you from Australia, and good afternoon or good morning to you wherever you are.
Goodbye.
Bye-bye.